Okay. Welcome back, everybody. We're beginning our first short week. So today we're going to be knocking out, hopefully, the gastrointestinal system. On Wednesday, we'll start with reproductive stuff. Then we have Easter break. So we'll finish that when we come back. Okay. So we've gone through the absorption of our different types of macromolecules that we're interested in. So we've talked through carbohydrates, we talked through proteins, we talked through lipids last time. We briefly talked a little bit about the absorption of vitamins, as well as some important ions, mentioning those. So we're continuing our little addendum of things to remember that your GI tract can absorb. So in addition to absorbing those macromolecules, absorbing vitamins, absorbing our sodium, our chloride ions, our potassium, we're also going to be absorbing and sometimes secreting bicarbonate. So a reminder, bicarbonate was important in our blood because it can buffer out acidity. Same thing is kind of happening in your GI tract. You have a lot of acidity moving through as we have chyme from the stomach full of hydrochloric acid. So we know that we need to neutralize that acidity in order to not basically burn our whole digestive tract. So bicarbonate is important. So bicarbonate is one of the things that's coming in our pancreatic juice, one of the big, long lists of things coming from the pancreas, as well as from some other places. Okay. So if we're thinking about bicarbonate, any bicarbonate that's moving through your system, we're going to start by trying to absorb some of that bicarbonate back into the system as we're passing through the small intestine. So it's in the duodenum that we have the acidic chyme coming in, also where we have pancreatic juices coming in, also where we have bile coming in. So that's really where we're thinking about the neutralizing of acid happening. So as we neutralize that acid, it does get converted into water and carbon dioxide that we can absorb. So this is happening in the jejunum. And then as we move further down our digestive tract, so as we move to the ileum, the last part of our small intestine, or move into the colon, we're going to see that there can be some bicarbonate secretion happening in these areas. So now that we've hopefully absorbed everything we need to absorb, we've done a little bit of fiddling with the contents, we may now be exchanging some bicarbonate, putting it back into the lumen of the tract as we exchange it to pull in a couple more chloride ions. Calcium is another thing that you should be absorbing. This will also be happening in the small intestine in the first parts, so in the duodenum and the jejunum. There's a specific brush border protein that is responsible for helping you transport that calcium into your body. So kind of like the brush border enzymes we saw when we were looking at protein absorption, we've got the same kind of thing happening for calcium. So we have a calcium binding protein, helpfully named there, that helps us transport that calcium into our epithelial cells. And then we have a calcium pump that pumps that calcium into the blood on the other side of your cell. We also want to make sure that we are absorbing iron. So we know that our body goes to some effort in order to recycle iron as we break down red blood cells. And that was one of the things we're trying to recycle from those heme groups. But we also need some dietary iron, right? Just recycling isn't enough for your iron needs. So we're going to be absorbing iron from our diet. So the way we do that is we need to bind iron to this molecule transferrin that allows us to physically move iron around our bodies. So in this case, it's your intestinal cells, so some enterocytes that are going to be secreting this transferrin, right, this molecule that we need in order to be able to grab on to iron. Okay, so it's going to be secreting it into the small intestine. It's going to grab some iron, and then that complex, which before we saw listed sometimes as T iron, so transferrin and iron bound together, is able to be taken in by the cells of your intestine. So they'd be binding together to a receptor, and then endocytosis would occur. So we're basically going to have that transferrin and iron bound to a little membrane protein, and then we're going to engulf it and pull it into the cell. So we can leave it inside our intestinal cells, inside those enterocytes, in which case we'd store it as this F iron bound to ferritin. Or we might keep that iron moving, move it into the blood. So we'd bind it to transferrin again, push it into your bloodstream, let it move around your cardiovascular system until it gets where it needs to go. For example, potentially to some red bone marrow that's actively making some new red blood cells. Finally, just a note, one of our big inputs of water into your body, water is super important, is from your GI tract, right? From the water you drink and from any food that has water in it, which is like most food. Okay. So in terms of the water in the GI tract, you're usually like taking in only about 2 liters a day, but all those secretions full of enzymes like the gastric juice, the pancreatic juice, actually add up to a fair amount of water, potentially up to like 7 liters a day. So we're going to need to make sure that we're absorbing that water back, right? So we're using it to secrete enzymes into the lumen, but we also want to absorb it back into our bloodstream, make sure that we keep our blood volume up, right? Don't make life too hard for your kidneys. All of that's going to be passive. So that water absorption is going to be happening a little later on. It's going to be following all these various solutes that we've been tracking throughout the system. So as we're absorbing the ions, the minerals, the vitamins, we're basically upping our osmolarity, right? So that pull on water. So the water is going to follow that pull as we've absorbed all of these many different components that your body needs. And that's as detailed we're going to go on the absorption of these other things that are very important for your body. Okay. So next we're going to talk about how we control digestion and how we move food through our GI tract. Okay. So we're going to start out by thinking about some hormones and some reflex pathways. Okay. So when we talk about gastrointestinal control, kind of in a big picture perspective, we're often going to divide where our signal for whatever response that we're looking at is coming from. Okay. So what do I mean by that? So basically, we're going to be thinking about, are you getting a stimulus to your head, so your cephalic phase, that's telling your body to release some hormones and change your digestion? Is this a signal coming from the stomach? That's going to be your gastric phase, that's telling your intestines to have a particular response. Or is this a signal coming from later on down in the intestinal tract, telling your stomach to do something or telling your brain to do something? So we're going to be thinking about different types of responses. And as we're thinking about that, we're also thinking about how far a signal needs to travel. So when we see these long reflexes and short reflexes, our long reflexes, we're thinking about reflexes that have to like travel on some type of autonomic nerve, right? Be it sympathetic or parasympathetic. Signals that have to get all the way back to your brain. Those are going to be long reflexes. Short reflexes are going to be kind of like when we've spoken about local control before or intrinsic control. Short reflexes are going to be things happening right in that general type of area. So like if you have a gastric phase stimulus coming from the stomach that's acting on the stomach itself, that would be a short reflex. If you have it going all the way through your systemic circulation and up to your brain, that would be longer. Okay. So each of these is going to be responsible in part for doing some hormonal regulation that's going to result in the release of these different enzymes from the different glands that we've been talking about. So cephalic phase control is going to be including thoughts that you might have about food if you're daydreaming, about some French fries, tasting food. your mouth is in your head, so this is still cephalic phase. Smelling food, these are all signals that are going to be originating from your brain and your central nervous system. And so we're going to need a longer pathway to get out to the rest of your body. We're going to see stuff going through your central nervous system. For gastric phase, we're really going to be thinking about events happening in the stomach itself. So we're going to be thinking about your stomach sensing, is there food in the stomach or not? How stretched out is the stomach? How acidic is the stomach? That's the kind of thing we're thinking about with gastric phase control. And that will have some influence on the stomach itself, but we have some hormones that get out into your bloodstream from there as well. Intestinal phase stuff, same thing as gastric phase, but now instead of stomach, we're thinking small intestine. So as we have receptors in the small intestine, being able to sense what's moving through the small intestine, those would be our intestinal phase stimuli and then reflexes. So as an example, I got a little scenario here where we have this person walking past a bakery and smelling fresh baked bread. I'll go ahead and read the question and let me know what phase of gastrointestinal control we're talking about here. Take a look. Okay, great. Yeah, so this is cephalic phase. So we've presumably all had this experience, right? Where you smell something you really like to eat and you start to feel your mouth dripping with saliva. You start to feel your stomach beginning to churn, right? So these are physical signals that this input, this smell, this thought, this sight of food is priming your body to start that chemical digestion process. So that's where we're going to start. Okay. So when we're thinking about digestion events that are happening in the head, let's think about that saliva. Right, so we know we have salivary glands in our head. We've seen some of them, right? Our parotid gland, our submandibular gland, our sublingual gland, right? So they secrete saliva that is able to both help us move food down through the pharynx and into the esophagus by lubricating it, as well as begin some of the digestive processes. We focused on our salivary amylase that starts to break down starch, so starting to break down carbohydrates in the mouth. Okay. So at the same time this is happening, as you're tasting your food and feeling the texture of your food in your mouth, there are receptors in your mouth that are sensing that taste and that texture, right? So we have mechanoreceptors. So think mechanical, so feeling that physical touch, that texture. And chemoreceptors in the mouth would be helping you taste the food, right? And going back to a variety of cranial nerves, back to your brain, carrying the sense of taste. Okay. One of the places that these signals are going when you're feeling and tasting the food in your mouth is to a region of the medulla called a salivary center, which is linked up to your autonomic nervous system. So it's going to be regulating parts of the parasympathetic nerves and parts of your sympathetic nerves. Okay. You also have some input coming from sight centers. So the food that you are seeing, any food that you're smelling, that's all information that's coming back to your brain. So usually coming to some part of the cerebral cortex, so that kind of outer part of the brain. Also then relaying some signals to this salivary center in the medulla. Okay. So if we've sensed with these cephalic phase stimuli that we have food in our mouths or food about to be in our mouths, we're going to see the stimulation of saliva production. So we're going to start to make more saliva so that you can use it to lubricate food that you might eat and move it down your digestive tract. Okay. So the parasympathetic branch of our autonomic nervous system, we usually think of as the rest and digest branch. Okay. So this parasympathetic part of our autonomic nervous system is going to be responsible for that sort of flooding of your mouth with extra saliva. So when we have parasympathetic activation happening here, that rest and digest activation, we're going to produce this watery, more kind of voluminous saliva as we prepare to eat food. During other parts of your day-to-day life, if you're not smelling food, you're not eating food, maybe you're really nervous, about to give a presentation, something like that, your fight or flight reflexes are being stimulated, aka your sympathetic part of the nervous system. You'd still produce saliva, but it's going to be very different in its composition. It's not going to be as watery. There's not going to be as much of it. It's going to be pretty heavy in proteins. It's going to be kind of viscous. So when you're producing that kind of saliva, you may feel that even if you physically have moisture in your mouth, you might be getting like dry mouth, this kind of feeling as you're getting nervous and you have this sympathetic activation in your salivary centers. So that's happening in your mouth. And then we're passing through food, down through our pharynx, into the esophagus, and down into the stomach. So the next thing we want to think about is how we're deciding how much acid to produce, right? So we know we're producing hydrochloric acid. So particularly those hydrogen ions we were thinking about, those protons coming out of the gastric pits. And also pepsinogen, so our precursor for pepsin. That's part of that beginning kind of workflow for digesting proteins. Okay, so these are also going to be linked up to your autonomic nervous system, but we're also going to have some more local hormonal regulation here. Okay. So when we're thinking about acid or pepsinogen, we're going to be thinking about parasympathetics as upping their production, right? So parasympathetic, rest and digest. You're producing more saliva, which is kind of watery, and it's starting to trigger this stomach acid production and this pepsinogen production. So when you think about your stomach grumbling, you can kind of think about that acid and pepsinogen that's getting ready to help you digest, particularly proteins, but also other things. Other things that we might be responding to when we're thinking about acid and pepsinogen. So we have gastrin, so that's a hormone that is being produced by the stomach itself from those G cells that were never pictured on our diagram, but do exist in the stomach when we're thinking about the gastric pits, as well as histamine, so that kind of inflammatory molecule that we've mentioned here and there, but never really focused on. So these are other signals that we would have for producing acid and pepsinogen. In general, we're going to be producing acid and pepsinogen at the same time, right? If it's appropriate to produce acid, right, that means you're trying to denature proteins, right? You're expecting there to be a bunch of new stuff coming in. Maybe you're killing off bacteria with that acid. Lots of different purposes of our stomach acid, one of which is to activate the pepsinogen. So it wouldn't be all that helpful to produce tons and tons of pepsinogen without producing acid, because that pepsinogen is in the inactive form. We need more acid in order to activate it to pepsin. So they're going to be linked. So some of the stimuli for this increase in acid and pepsinogen secretion in the stomach are going to come from your head. So we're going to have cephalic phase regulation of the secretion of these two molecules of interest. So if you see food, you taste food, you smell food, you chew food, you swallow food, those are all signals that you're going to be getting from your head that food is about to hit the stomach. So when we're getting those signals, they're going to be activating the parasympathetic nervous system. That parasympathetic part of the nervous system made you make all this watery saliva, and it's also going to be stimulating this increase in acid and pepsinogen in the stomach, partially through the stimulation of gastrin, which is one of our hormones related to the stomach. So here we see a kind of flow chart of how this is working. So here are our various cephalic phase inputs here. So seeing food, smelling food, tasting food. So you can see that this is increasing how much parasympathetic activity is traveling out to your body. So parasympathetic activity, not under voluntary control. This would happen to be traveling out on your vagus nerve all the way down to the nerves going to your stomach, right? So some of that is going to act directly on the parietal and chief cells that are in the gastric pits of your stomach, increasing acid and pepsinogen secretion from each of those cell types respectively. And we have kind of this additional effect that's going to be increasing the acid and pepsinogen production, because we also have this kind of side route that's also ultimately increasing acid and pepsinogen. So here we have those G cells from the stomach producing gastrin. This is putting gastrin into circulation. So in your bloodstream, you're getting gastrin, this hormonal signal. And then ultimately, that's also acting on those parietal and chief cells to help you produce more acid and pepsinogen. So we've done the same thing 2 different ways here, basically. But it's not just cephalic phase control that's happening when we're thinking about the stomach. So when we're thinking about salivary centers, mostly that's just going to be stimuli coming from your head. But now that we're down in the stomach, we're going to actually be thinking about some gastric phase control, so some actional signals coming from the stomach itself. So your stomach also has receptors for sensing what's inside, as well as how big it is, how stretched out the gastric rugae are, so those accordion folds in the stomach. So the way that your body knows from this gastric phase perspective that it's time to make more acid and pepsinogen is it's noticing, oh, the stomach is kind of big. That's the distension part, right? So it's bigger, must be something in it. And the other piece is that we're sensing those things that we want to start actually chemically digesting with enzymes. So we're going to be noticing that we have proteins in the stomach, that we have peptides, amino acids, so smaller pieces of proteins, indicating that it's time to be producing pepsinogen, activating it with acid so that we can continue this digestive process. So there will be some further reactions in response to this. So we're going to have some short reflex pathways, so local affecting the stomach itself, as well as some longer reflex pathways that end up communicating kind of to your whole body. Okay, so we're going to be triggering gastrin, acid, and pepsinogen release, just like we were with the cephalic phase stimuli, just kind of coming from a different signal here. So we're still going to see the end result of our parietal and chief cells producing more acid and pepsinogen. But now we're thinking about our gastric phase control. So our stimulus is now those proteins and some broken up proteins in the stomach, as well as how much our stomach is distending, how much it's stretched out. So we have chemoreceptors sensing that chemical composition of the stomach. So that's the protein part. And mechanoreceptors feeling that mechanical stretch, that distension. So we're going to have some reflexes going directly to those parietal and chief cells. We're also going to have some longer reflex pathways that involve first locally producing this gastrin, but then sending it into your bloodstream. It swims around, gives signals to other parts of the body, but ultimately comes back and helps you produce more acid and pepsinogen. Okay. So far, we've had both our cephalic phase and our gastric phase telling our stomach that we've got food incoming, or we have food right here. We need to digest it, so we need pepsinogen to break down proteins, which means we also need acid to activate the pepsinogen. But you don't want your stomach kind of going runaway on the acid and pepsinogen secretion. You do also want this to stop when you don't have food in your stomach, and stop as you've passed the chyme into the duodenum, and now you have all that food moving through your small intestine and your stomach's kind of empty now again. So we have some ways that we can inhibit the secretion of acid and pepsinogen as well. So we're thinking of this as like the turning off the signal kind of portion. Some of that is still gastric phase control, right? So we had gastric phase control that turned on this production of acid and pepsinogen. But as we see that food leaving the stomach, so exiting through the pylorus and that pyloric sphincter and going into the duodenum, this is now removing some of that stimulus that we had for producing acid and pepsinogen. And we also see that now the stomach, when it has less food in it, is going to be building its acidity, right? So while we're still producing acid, now there's going to be relatively more and more acid as the food has left. And that's going to also inhibit the production of gastrin, which is our kind of like local hormone for the stomach, which is going to help us kind of turn off these gastric phase signals for the stomach to produce acid and pepsinogen. It's not just the stomach noticing that it's emptying and having this food leave, though. We also have signals kind of looping back from our small intestine, looping right back to the stomach, telling your stomach that this food has now moved on to the intestine, right? So the stomach is noticing, hey, I'm not sure the food's here anymore. Your intestine is going to be telling your stomach, yeah, we have the food. You don't need to worry about it anymore. So this is where our intestinal phase control comes into play. So helping to kind of turn off this signal for acid secretion and pepsinogen secretion. So as food moves into the duodenum, so that first part of the small intestine, this is going to increase its osmolarity. So now we have a bunch of stuff in the duodenum that is not water. So we see an increase in osmolarity. Some of the stuff that we're passing in probably has triglycerides in it, has lipids in it, has fat in it. So that's something we can sense in the duodenum. And it's also going to have acid because that chyme is part stomach acid, right? So we mixed our food together with stomach acid. So that's an acidic fluid that's dribbling into the duodenum. Finally, your duodenum can also notice of the duodenum as it physically increases. Okay. So we're going to have some signals coming from this intestinal phase control that are looping back to the stomach, inhibiting acid and pepsinogen secretion, as well as affecting your kind of larger system when it comes to digestion. Okay. So we have cephalic phase and gastric phase, turning on production of acid and pepsinogen. Then we have gastric phase as the stomach is kind of emptying, noticing that increase in acid, removing those stimuli for secretion. So that's kind of tamping down on acid and pepsinogen secretion. Plus, we're getting these signals from the intestines saying that the food is now in the intestines. So we don't need to produce all the stomach acid anymore. Okay. So now that we have our food in the duodenum, right? So we've passed from the stomach to the duodenum. It's now important to think about the pancreas, because our pancreatic juices are going to be coming into the duodenum through the pancreatic duct, the ampulla levator, and then the sphincter of Oddi. And we're also going to be thinking about bile, right? So bile coming from the liver, maybe also the gallbladder if we've stored some extra bile, ultimately coming out the common bile duct, again, to this same region of the duodenum. Okay. So we're going to be thinking first about the pancreas stuff. So when we think about what the pancreas looks like, what this gland is like, hopefully you've gotten a peek at it in lab. It's this kind of dense glandular tissue, kind of tucked behind your stomach or kind of cupped a little bit by the edge of the duodenum. And we're going to have cells that are responsible for secretion of enzymes, as well as cells that are responsible for secretion of bicarbonate when we're thinking about what this is doing for digestion. The pancreas does have other jobs as well. Okay. So when we're thinking about the cells that are making our enzymes, we're going to be thinking about the acinar cells. So the acini are like little balls, little clumps of gland all together. And they're going to be responsible for a small volume of the secretion. So not that much fluid, but really important because that's where our digestive enzymes are. We're going to have some water in this. We're going to have some electrolytes coming from the acinar cells. The duct cells, so physically lining the duct as we are moving towards the duodenum, are also producing fluid. So they're going to be producing more fluid by volume, and it's going to be full of bicarbonate, right? So this is that let's buffer out the acidity part of the system. Okay. So we're going to have two hormones that we're thinking about helping us regulate the bicarbonate stuff and the enzyme stuff. Okay. So CCK and secretin are our two signals we're thinking about when we're thinking about how the pancreas knows whether we should be producing more enzymes, whether we should be producing more bicarbonate or not. Okay. So technically, the regulation of our enzymatic production, so the pancreatic enzymes, versus the regulation of the bicarbonate coming from the duct cells is separate, right? So theoretically, these are being regulated independently, although we see them often operating at the same time. But they do have their own kind of signals associated with them. The pancreatic juice, as a result, that's coming into the duodenum can have different amounts of enzyme, different amounts of bicarbonate, because we're regulating these separately. Okay. So our first stimulus, or our first kind of hormone we're thinking about here, is CCK. So CCK is going to be responsible for enzyme production. So the way it's going to do that is it's going to act on the acinar cells, and it's going to tell them to secrete more enzymes. That's its main job, right? So on this list, the big things are their main job. Okay, so CCK is helping us make enzymes. Secretin is telling the duct cells to make bicarbonate, right? So those are their two top line jobs. And when we say they're regulating independently, that's really what we're talking about. There are some details here, though, that lead to them being a little bit linked. Okay. So CCK is mostly telling your pancreas, telling the acinar cells to make enzymes, but it's also a little bit of a stimulus for the release of bicarbonate, right? So smaller effect, but still there. And kind of more importantly, it does something we refer to as potentiating the effects of secretin. So it basically like makes the duct cells more responsive to secretin. That is the chemical that is telling those duct cells to release bicarbonate. So it has kind of a amplifying effect on those secretin effects. The opposite thing is true for secretin and its effects on CCK and enzymes. So secretin's main job is to tell the duct cells to secrete bicarbonate, but it is a small stimulus for enzyme release, and it's definitely potentiating the CCK as well. So it's amplifying that effect by making your acinar cells more receptive to CCK. So we've got two main signals, two main hormones involved here. They each have their own big main job, but they also help the other hormone do its job. Okay. So for each of those, we have signals that we'd be tracking from the GI tract. Okay. So if we want to release CCK, CCK is the one that's telling our acinar cells to make enzymes, right? Okay. So we're going to see stimuli related to that, increasing the release of CCK. Okay. So enzymes that we're going to need for breaking down food, we're going to need lipases to break down fats. That's one of the important things happening with our pancreatic juice, right? So fat in the duodenum is a stimulus for CCK release, right? So when we have those fats, we know we want to release some of that stuff. Amino acids are also going to be one of our stimuli for CCK release because we saw that a whole slew of enzymes coming from the pancreas are for the digestion of proteins, right? We had a variety of proteases coming from the pancreas. So when we sense amino acids in the duodenum, that means it's time to continue the digestion of protein. So that's also going to be a stimulus for the release of CCK. For our secretin, its job is to stimulate duct cells to produce bicarbonate. So its main stimulus is going to be when we sense a bunch of acidity in the duodenum, right? We want bicarbonate to help us buffer out that acidity to make sure you're not burning your insides. So when we notice extra acidity in the duodenum, we release secretin, tells those duct cells to make a bunch of bicarbonate. So here's what some of that would look like a little bit. All right, so if we sense acid in the duodenum, the reason we're sensing that acid in the duodenum is because we've started passing chyme from the stomach into this first part of the small intestine, into the duodenum, right? So that acidity is coming from our stomach acid, and we have some chemoreceptors that are now going to detect and respond to that. They're going to send off a bunch of reflex pathways that we're not going to be specific about where exactly they're going or how exactly they're working. But our end result is that we're going to come into the endocrine cells of the pancreas, so the ones that do hormones, and we're going to be producing secretin. So that's our secretin, which is great because this is a response to acidity. So now we're going to have secretin running around in our bloodstream and our plasma. It's ultimately going to come into contact with those pancreatic duct cells, having us make some bicarbonate, which is going to go through our duct, out our pancreatic duct into the duodenum, where we're going to neutralize that acid, which means we've now turned off our signal. So once there's no more acid in the duodenum, we turn everything off, right? So this is a negative feedback loop at the end. So these hormones that we're looking at are also helping us regulate bile secretion. Okay, so we're thinking about the pancreatic enzymes in the pancreatic juices. We're thinking about the bicarbonate coming from the pancreas, but we're also thinking about bile coming from the liver and potentially coming from the gallbladder if we have extra bile stored there. Okay, so the hormone that is responsible for that initial bile secretion, so like making the bile and the bile salts, is secretin. So secretin is talking to the liver, telling the liver to make more bile. CCK is helping us release that bile that we've made. So we don't need to just make the bile, we need to actually get as much bile as we can into the duodenum to start emulsifying our fats so that the lipases from the pancreas can start to break them down. So that's CCK's job. So our gallbladder is basically just storage for extra bile. So CCK is going to tell us to constrict that gallbladder, contract it, so that any bile within it is heading out through our common bile duct into the duodenum. And at the same time, we want to make sure that we are opening the hole into the duodenum. So the sphincter of Oddi is a tiny little circular muscle right at the entrance of that bile duct and the pancreatic duct into the duodenum. Right, so when we relax it using CCK, that means that the bile is just going to flow nicely into the duodenum. So we have these things all kind of working together. So let's scroll back, right? So we had our CCK helping our pancreas make enzymes, including lipases and proteases that are going to help us break down lipids and proteins. We have secretin helping us make bicarbonate so that we buffer out acidity of the chyme coming from the stomach. And now that we know both of these are also helping us make, in the case of secretin, and then release, in the case of CCK, the bile and the bile salts that help us emulsify lipids so that we can actually digest them with those pancreatic enzymes. So that's all working together. So here we can see some of what's going on when we're thinking about that bile, right? So one thing we have different things happening in the duodenum that we're sensing, right? So when we sense acidity, we produce secretin, right? So one thing the secretin does that's not on this diagram because it's not about the bile, but it's making that bicarbonate, right? That's why we're linking this up to that acidity signal, right? But now that we're focusing on bile, we know that the secretin is also helping us make bile in the liver. On the other side, we know that CCK is responsible for telling the pancreas to release enzymes. So our signals for CCK production are related to that. So proteins and fats, their presence in the duodenum is when you know you need CCK for those pancreatic enzymes. And along with that, to fully digest our lipids, we don't just need the pancreatic lipases, right? We don't just need to enzymatically break down the fats. We also need to emulsify them. So we need to break up those big fat drops into smaller little droplets, right? So we need that bile. So we're getting extra bile from the gallbladder, and we're relaxing our tubes, sending that bile into the duodenum. So the end result is that we have more bile in the duodenum. There is some bicarbonate here, so that's helping us neutralize acid again. And we have those bile salts for the emulsification of fats so that we can do our lipid digestion. Okay. So finally, the last little bit that we're going to go through here, which I think is hopefully pretty easy, is thinking about the movement of the GI tract. So we mentioned that we have muscles in various places in the walls of the GI tract, right? So because we have these muscles, right, we have the muscularis mucosae in that mucosal layer, that inner layer. We also have the muscularis externa, so these big, larger layers of muscle farther out. This means that your GI tract can move, which is great. You want to keep moving our waste products, basically, after we've done our absorption, down the tube and out the body. Okay. So when we think about GI motility, when we think about the movement of the GI tract, we're primarily thinking about the actions of those larger muscles in the muscularis externa. All right, so this kind of third layer here. So we saw that there were circular layer muscles and longitudinal muscles here. So these muscle layers are going to be helping us to mix the GI tract contents together. So scramble it all around, hopefully scramble it enough that it all comes into contact with the walls of the intestine so that you can digest and absorb. And then we're also going to want to propel the GI tract contents forward. We don't want everything just sitting like a rock in your duodenum forever. We want it heading all the way out the small intestine, all the way through the colon and out your body. So when we think about this, we're going to be thinking about that muscularis mucosae, so that big muscle layer in your GI tract, as having kind of baseline electrical rhythm, right? So kind of like when we were talking about how our pacemaker cells in the heart just naturally have a bit of a rhythm, the same thing is also going to be true here in the GI tract. So we have these natural waves of contraction coming from these cells themselves. So when we refer to the two types of movement, we're going to divide them based on their function. So we're going to be dividing them based on whether we're moving the contents forward, and that's peristalsis. So moving stuff out of your body, it's happening by peristalsis. So we're thinking like a snake eating, right? That vision is a mental vision of peristalsis on a very visible kind of way. And then we have segmentation. So segmentation is going to be helping us mix the contents of the GI tract together. So we'll take a look at each of those real quick. Right. So here's peristalsis. So by definition, right, what we're tracking is, are we moving food forward, which we are, right? So here's our bolus of food, moving forward, moving forward, moving forward, right? Peristalsis is moving this out. Okay, so the way this happens is we're basically like pinching behind your ball of food, right? And you can imagine how pinching something does kind of move it forward a little bit. The other thing we're doing is kind of progressively lengthening the muscle behind and then contracting the muscle forward. So think for that part with the lengthening and contracting, think about like trying to get a hoodie string back out the hole if it's slipped up in the laundry, right? That's what you're doing with these longitudinal muscles, right? So behind this, here we see that pinching off, right? And we're going to stretch out the longitudinal muscles this way. It's going to be long and skinny. So this ball doesn't want to move backwards. And then in front of this ball, in front of this bolus of food, we're relaxing the circular muscle. So this part of the tube gets wider, like side to side. And then the longitudinal muscle contracting would have the effect of like, this is where you like scrunch something, right? So that as you move it forward, right, it moves farther down, basically, is what's happening here. So we have these two arrangements of muscles, the circular muscles and the longitudinal muscles, both kind of working at the same time together to get this to happen, with the ultimate goal of moving from that small diameter area behind the bolus of food towards this larger one in front. And that's really all we're thinking about when we're thinking about peristalsis. Segmentation is particularly the responsibility of the circular muscle layer. And what we're going to be doing here is usually something that we're doing in the small intestine specifically, because that's where our absorption of nutrients is happening. So what we're going to be doing, like if you think about food, let's move through this tract, right? So this food came in first, this food came in second, and so on, right? We're going to be mixing this back and forth so that all the contents gets mixed together, trying to jostle it up against the walls. And this happens basically by alternately contracting segments. So you can think of your intestines as having kind of some little boxes within them, so subswellings within them. So if you contract this one and contract that one in a kind of alternating rhythm back and forth, you end up mixing together the intestinal contents. So this is also how we're mixing together the chyme, so that food and stomach acid kind of mix together. Okay. And that is it for our digestive system. So next time I see you, we'll move on to the basics of reproduction and the reproductive system.